Hi, Dr. Brian Kaufman, a family doctor and CLL patient myself, and I'm the medical director of the nonprofit CLL Society here at ASH 2017 in snowy Atlanta.
I'm Dr. Matthew Davids from Dana-Farber Cancer Institute and Harvard Medical School in Boston, and I'm a CLL specialist there.
So it's always a challenge to pronounce the names of new medications, but Umbralisib, did I get that right?
Exactly right.
You've done some research on how that drug works and the pharmacokinetics. Could you share that with us from a patient's perspective and how it might be helpful?
Sure. So patients may be familiar with Umbralisib. This is an approved PI3 kinase delta inhibitor for patients with CLL. It's a very effective drug, but it also has significant toxicities that tend to be immune mediated. So Umbralisib is a next generation PI3 kinase delta inhibitor, and we think it has a differentiated safety profile compared to Umbralisib. So the point of our analysis was to try to show that with a substantial amount of data. We were able to look across the Umbralisib early phase development program and pool together 350 patients treated with Umbralisib. And we found just what we expected, which is that the rates of colitis and transaminitis, liver inflammation, are much lower than what we've seen with Umbralisib. And we have pretty good follow-up now with many patients out 1 to 2 years on Umbralisib, and that's a period where we'd expect to see toxicity if there were going to be some.
And do we have any idea why this is different than the experience with Umbralisib?
So this is an active area of investigation. The drug does hit PI3 kinase delta. We have kinome scans that suggest that it's very selective for that target, but interestingly it hits another protein called CK1 epsilon, which we think may have effects on regulatory T cells. So there's some interesting animal data to suggest that that might actually be one reason why it mitigates the toxicity of the drug is through this regulatory T cell effect, but this needs to be confirmed in humans.
Well, I'm excited about us having another option because the PI3 kinase pathway is clearly an active and important pathway in CLL. Any final thoughts you have that you wanted to share?
I think the other thing I would say is that as we put together combination regimens of novel agents, we want to use the agents that have the best toxicity profile. So we're currently running a study of Umbralisib with ibrutinib, and I think that's a promising study given the toxicity profile of these drugs.
Dr. Davids, as always, thanks so much for the research you're doing. Thank you.
Thanks.